Do you think you've spent less during lockdown?
Oh, enormously. I don't know what I spent it all on, Michael.
I spent so much money doing renovation on my house, so I actually end up being a lot more expensive. Yeah, exactly.
Yeah.
Yeah.
People have been doing a lot of nesting.
I've done a fair bit. I've done a bit of, I've re-turfed my lawn on the weekend.
Oh.
Hi, and thanks for joining us at Phacilitate Talks. I'm Michael Adeniya, the Portfolio Director at Phacilitate.
And I'm Adeniya Davies, Founder and Chief Executive of Dark Horse Consulting.
And if you're joining us for the first time, Phacilitate Talks is a socially distanced talk show for the advanced therapies community, where Anthony and I are joined by two guests to discuss the big issues in cell and gene therapy and what our audience are dealing with right now. But there's a twist.
But there's always a twist, isn't there, Michael?
That's right, Anthony. And the twist is, rather than us ask all the questions, we invite our guests to ask the questions. So it's no hold bars, and we discuss whatever comes up. And if you enjoy the show and like the format, please rate us on Apple Podcasts, subscribe on YouTube or Google Podcasts and all of the various podcast platforms out there. And you'll never miss an episode. Also, if you love everything that Phacilitate do, then why not become a member and get a whole bunch of benefits, including content straight to your inbox, discounts, special invitations, and the ability to be a part of an exclusive group. And you can sign up at www.facilitate.co.uk/subscribe. So on to today's show. And this week we've got two fantastic guests joining us. Our first is joining us from the suburbs of Philadelphia. She's been with GSK for 14 years, and her career has seen her develop biopharma and cell and gene therapy products from target to post-launch. She has responsibilities ranging from 3 to 5 year business strategy, creating high-performance teams and talent development, technology and partner management, as well as R&D strategy. She has a PhD in chemical and biomolecular engineering from Georgia Institute of Technology, specializing in retroviral and lentiviral gene therapy. She's also a Women in Advanced Therapies mentor and has been mentoring young women in the cell and gene therapy space through Phacilitate's program for 2 years now. She loves nature, cats, and spending time with her 7-year-old daughter. Please welcome Delfi Krishna, Director of Cell and Gene Therapy Platform, R&D Strategy, Portfolio and Operations at GlaxoSmithKline. Thanks for joining us, Delfi.
Thank you for having me.
And our second guest has a career that spans over 25 years with one company, and that company is the global leader in diabetes research. He heads up their global diabetes research with one objective: to find a cure for type 1 diabetes. Now, he obviously has several million people eager for him to be successful in this. One of those being his daughter, Vita, who has type 1 diabetes. I'm delighted to have joining us from his vacation home in Denmark, which I understand the weather is a little bit choppy today. It's our corporate vice president and head of stem cell R&D at Novo Nordisk, Jacob Peterson. And Jacob, thank you for joining us while you're on your vacation as well. We really didn't want to miss the opportunity to have you with us.
No, absolutely not. This is a big pleasure for me. And we actually have, as you said, the worst storm ever in 50 years. So it's nice to be inside and be talking to you guys instead of being out where I normally am when I'm in my summer house.
Wow. And is this the first day of your holiday, or?
Second day.
Second day in, I see.
Yeah.
You should have gone to England on holiday. The weather's always nice there, right, Michael?
Well,
yeah, right.
Yeah, Wales, the Welsh weather is probably not that dissimilar to where you are, Jacob. And I was saying earlier, so yeah, I was laying my lawn on the weekend, and thankfully, because of the Welsh weather, I don't have to drain my sprinkler to keep my grass wet. So for some people it's good, For some on vacation, not so good.
Yeah, I'm looking forward to a little bit of sunshine, I must admit, but it's going to come in the next few days, so that's good.
And speaking of, thinking of holiday weekends, it's obviously just past Independence Day weekend. What's that been like in Philadelphia, Delfi?
It's been really, really hot, close to 40 degrees, and quite humid as well. But everybody's out and about with their masks on, and it's just a pleasure just to enjoy nature at this time.
Nice.
With what's going on everywhere.
Nice. So you were able to get out and spend some time outdoors over the weekend?
Absolutely. This summer, I taught my daughter how to ride a bike. So we've been biking trails. So it's been a lot of fun.
Oh, that's brilliant. And I think Jacob and Delfi, are your daughters around the same age?
My daughter is 7.
Yeah,
well,
so
I have
is mine.
6 kids. I have 6 kids, so it depends on which daughter you're. So 25, 7, and 5.
Oh, wow.
Yeah.
Brilliant. Brilliant. Well, are there any twins amongst those?
No.
Wow.
So I have a daughter that's 27, a boy that's 26, a boy that's 16, a boy that's 13, adapted from South Africa, and then 2 girls that's 7 and 5. And Vita, the one who's 7, she's the one with type 1 diabetes.
Oh, wow. And yeah, I mean, we're all very much a family orientated group here. Anthony and I have, Auntie, as you know, I've got one on the way, and my eldest is around the same age as your eldest. Is that right? I think there's a few months apart. Is that right? Yeah. Mm-hmm. Yeah. Oh, good. So without further ado, I reckon it's time to jump into our first question. And we normally like to allow the guest to go first. So over to you, Delfi. Would you like to go first with your question?
Yeah, absolutely. Thank you. So as our folks and our family members at Phacilitate and in the industry may know, GSK has a focused portfolio in cell and gene therapy on CAR, TCR, ex vivo autologous viral modified products. And we have a lead asset in the clinic, which has been in license from Adaptimmune. And then we have some others which are entering the clinic. So my question is around the good old problem about cost of goods. And in addition to that, because it's ex vivo autologous vein-to-vein time. And what I would like to love to talk about today is what approaches are we in the industry that our experts are seeing being used to reduce cost of goods and vein-to-vein time.
Nice. I'm going to hand over to Anthony to kick things off and share your thoughts. Really good question.
Yeah, no, that's a great question, Delfi. And it's something that of course, we've been borderline obsessed with over the years. And it's always been a concern for me that I think the phrase I used in Miami this January, Michael, is that I'm concerned that the field is sleepwalking into a COGs crisis by inattention to COGs. Autologous COGs has issues of its own. I think allogeneic medicine is great, and I think we'll hear a bit more from Jacob about that. But I think there'll always be a place for autologous medicine. And you said something very interesting there. I think when you said that we should address the cost of goods and vein-to-vein time, because in a very real sense, when you're doing labor-intensive manufacturing and skilled labor-intensive manufacturing, time is money. So optimizing COGS might be very much impacted by reducing that time period as much as any critical raw material or something. At GSK, what's more important? Is it time or money?
I think it's both. And obviously, as you're saying, we don't want to walk into a disaster of, cost of goods and vein to vein time. So we've taken a very data-driven approach to both of the things in terms of, and I think Dark Horse has also published several papers on this. So we've taken a look at all of them, and we're very grateful for them. Really modeling-based approach, taking all the different processes, putting them in models, using data, simulating, thinking, is it labor, is it consumables, is it factory utilization, what is it? And with ex vivo autologous, we've really tried to align our technology investment strategy with what the models are telling us in terms of, cell processing, trying to understand what exactly is the kind of cell that you have to go after, because we don't really have a very good handle on that. So defining that, understanding that, phenotyping that, correlating that to function is a big effort that we are trying to get into. In addition to that, we think analytics is also a big part of how we deliver these, especially advanced analytics, rapid analytics, using less and less tests or moving them upstream, trying to do two-stage release, these kind of things, where time is money, all of that, we are actively looking into that. For viral vector, we had an early breakthrough, and we were able to follow we were able to patent a technology in which we are able to use a monoclonal antibody-like platform where we have a stable cell line, and we sort of put our therapeutic insert along with everything else for the lentiviral particles and make packaging cell lines and harvest viruses. So that's been quite successful in terms of bringing the COGs down, in terms of reducing the problem with variation of transient transfection and all of that. But we have both approaches in play depending on what our projects are. So that's sort of our COGS approach.
And if you had to put your finger on one thing, would it be the critical raw materials? Would it be equipment? Would it be QC overhead? Would it be human capital?
Yes, I mean, what our models have shown us is essentially two big things. One is the utilization of the facility. So wherever you are, that's the biggest thing. And I believe your models have shown something similar, right? And then the second big thing is human capital. So
Yeah, we completely agree with that. I think you referred to my colleague Katie Spink and Andrew Stein's article in 2018, which was loosely based on the Yes-Carter process, but trying to be a bit more generic. And I think one of the most surprising things to drop out for us was the sensitivity of the analysis to plant occupancy. Obviously, that's related to having highly skilled, expensive people sitting around as well. But that the level of sensitivity to that really surprised us.
Yep, absolutely. Yep.
Yeah.
And in autologous medicine, I think it was Nick Leslie from Bluebird who first articulated this, and I didn't quite understand it at first. He said, failure is not an option. And I just thought that was some sort of Nick buzzword, but he really meant it, because for each individual patient, failure, there is no medicine cabinet to go to pull out another vial of drug. And there may not even be another opportunity to do manufacturing. So in a sense there, not confusing expensive with value for money. I think that a highly reliable and robust manufacturing process may be kind of worth more, where lot failure is not acceptable. So, Jacob, a good friend of mine and someone I think we all know well in the field is Stefan Miltenegg, who's highly. We know what they make. Okay, we don't need to pretend we don't. And he started a presentation once a number of years ago by saying that he was, for many reasons, including cost of goods, a great fan of allogeneic medicine. And his comment then was, just one problem, the human immune system. So how is that argument? How was that argument 20 years ago at Novo? How was it 10 years ago? And how is it today? And is allogeneic medicine going to solve all of the problems around cost and personalization?
That's a great question, Anthony. At least you can say that's the route that we as a big pharmaceutical company has chosen to take, that we want to go after allogeneic cell and use that for cell therapy, because we believe we better can control batch to batch. But I think what Delfi said about analytics certainly also go for allogeneic medicine, because you have cellular products, and if they are not the same, then and you can show that they are the same, just like you have to do in autologous medicine, then it's not good. So we spend an enormous amount of time on our analytics and release criteria, but certainly also can recognize all the issues in terms of the COGs that you guys are speaking about, in terms of qualification of raw materials for our type 1 product. I think we have 4 persons full time just doing qualification of raw materials, so it's a humongous undertaking. And what we try to reduce COGs of good, besides being very diligent in planning the manufacturing, because that is very important to have run at full capacity as much of the time as possible. That is actually to see in the differentiation process, are there any outliers in terms of factors and chemicals that cost a lot of money, and then see if we either can produce it ourselves or make agreements with vendors so to bring costs down. Because normally our experience in manufacturing is that it's a couple of compounds that are very, very expensive, and that can be addressed, especially if you can produce it yourself. Then your second question, and that was the immune system and allogeneic cells. I guess we, like everyone else, are trying to make cells, embryonic stem cells or iPS cells that the immune system cannot see, by removing class 1 and class 2 and introducing something that inhibits NK cells. Everybody's doing it along the same lines, but nobody has actually so far shown that they can do that successfully in man. And that's going to be really, really critical, I think, for the overall long-term success of using allogeneic stem cells in your differentiation protocols, that you actually can demonstrate that you can produce cells the immune system does not recognize. What we are going to discuss a little bit later is devices, because you can also put cells in devices, but that's a little, I don't know if it's more complicated, it's a different avenue at least, but cells, making a stem cells is going to be really important. Also, hopefully later on, to be able to replace some of the autologous cell therapies, so you can go up in volume and scalability and consistency. So it's going to be important with a cell line like that.
And just one thing you mentioned, Delfi, around capacity, like utilizing the available space and the capacity that you've got. In terms of the modeling you're doing around that, is any of it to do with the type of devices that you use within that space? Whether it's isolators or what kind of incubation spaces that you use and so forth.
Yeah. So we have. Yeah. Oh, sorry. Go ahead, Delfi.
No, please go ahead.
No, women's first, please.
Okay. So yeah, absolutely. I mean, whatever sits within that facility, our central aim is to close it, automate it, and reduce as much human capital as possible without compromising quality and all of those good things. So our approach has been, wherever several companies I have noticed have taken an approach of joining a lot of unit operations from end to end, and that will require operator input and very skilled, highly skilled operator input. I mean, it's no secret GSK has a collaboration with Miltenyi and we've invested in the Prodigy platform. But absolutely, it depends on how much skilled labor is required for whatever is in that facility, to what you can, how much lower you can drive down the COGs. And once you've settled on an automated solution, it's about really understanding what is it that you want in your cellular drug product, and how does that correlate with safety, efficacy, outcomes.
Yeah, I think it's all about making the right cells. I think we've heard that from both of you. One of Bruce Levine's just amazing studies was the one where there was a clonal expansion from essentially a single cell in a CAR T product, which was responsible for the efficacy of that particular dose. And it's just a matter of picking the right cell, and you don't have to do much manufacturing at all. On Jacob, on your side, it's about, I guess, getting to the right population, because you have more of a, you're delivering the mass action in the drug product rather than in subsequent proliferation of the cells to such an extent. But the same, the need to exquisitely phenotype your product is critical for allo and auto alike.
Wow. Well, plenty of food for thought.
No, absolutely. I think that, and I guess that's the same as Delfi is saying, that the analytics is of vital importance. Coming from a protein and peptide company where you can do an HPLC and then you can look at impurities, and maybe if you go crazy, you can do a cellular assay in certain regions of the world where that's demanded by authorities, but that's basically it. I think the complexity of analytics of cellular products is just enormously higher and requires much more skills and multiplexing between different disciplines, from RNA-seq to a lot of other disciplines in order to be sure that the cellular product you have is the right one. And especially when you are not working with autologous cells, you need to be absolutely sure that you don't have any pluripotent cells that are left, because those can cause, of course, teratomas. So analytics for that is also a big distinguish whether you need a lot of cells, because then your analytics really need to be able to detect 1 in a million cell, or maybe like for retinitis pigmentosa or dry AMD, where you need hundreds of thousands of cells. Your analytics are not that demanding in terms of detecting unwanted cells. So it's actually a big issue for us, and something we are spending a lot of time on.
Well, I hope that gives you plenty of food for thought, Delfi. And thank you for the question. So next, it's over to you, Jacob. And as Anthony said, the topic of devices was something that is quite close to you. Do you want to tell us more about that?
Yeah, I mean, we started out at Novo more than a decade ago looking into type 1 diabetes. And at some point in time, we were doing both a device to protect from autoimmunity and to oxygenate the cells, and then developing the cells. But we basically said for basically a number of years, let's focus on getting the cells right. And now a number of companies, including Novo Nordisk, have kind of gotten to a point where we can make the cells. And I wouldn't say people have forgotten about the device, but now I think everybody in the field is struggling finding a device where you can put the cells in so they are protected from autoimmunity and rejection, because we don't yet have a stem cell. And also that because the beta cells that you use to produce insulin to cure type 1 diabetes is an oxygen demanding cell. So you also need to have a device that either fast become vascularized so it can supply oxygen, or that you supply oxygen from the outside. And that is probably as complicated as it gets in terms of developing a device. And I know that we and many others are struggling developing devices where the cells can survive, because in type 1 diabetes, you also need a huge amount of cells. So you need basically 1 billion cells in a very small area. So it's really, really complicated. Parkinson's disease, you need, that we are also working on, to deliver the cells in with millimeter accuracy in a specific region of the brain. Stroke, you also need to develop the cells into the peri-infarct region. and so on and so forth. Epilepsy, you also need a precise delivery. Chronic heart failure, you also need to be able to deliver the cells so they're not pumped out immediately again. So I think that many of our collaborators, they are looking at the differentiation protocols. They can make cardiomyocytes or beta cells, and then they kind of think, well, now we're almost there. But I can tell you, then that's where all the trouble starts, because then you need to find a device, and that can actually take years to develop. And the good thing is that if you get it right, it can also be a competitive edge. So it's a complicated area.
Yeah, no, that's so interesting. And I remember even further back, Jacob, when I was working on a spinal cord injury product. Again, that was also an embryonic stem cell product. You'll remember from the Geron days. And there, yeah, the dose, again, a small dose, relatively small dose, had to be delivered in the peri-injury area, as you say. You know, get it too close to the injury and it's just toxic. Get it too far away and probably not going to see an effect. And this was being delivered intrathecally to a patient who, you know, by the way, is breathing, so their spine is going up and down to a certain extent. And I think, you know, there was a lot of device engineering to solve there, and I completely agree with you. That taught us the hard way the value of excellent dev eng. And these are combo products. You know, this is a situation where, in a way that's perhaps not the case for, you know, Delfi's products, where the device is just and its delivery is an intrinsic part. You know, luckily, I think transfusion medicine, you know, a decade or two ago solved many of your problems, Delfi. But you know, chip in if you think there's anything unacknowledged there. And I think, you know, you got the ultimate set of conflicting variables, Jacob. I mean, I think something, unless I missed it, you didn't mention that, you know, the little matter that your cells have got to be able to sense glucose and release insulin and get those molecules in and out. But I'm guessing you don't want too much complement and too many antibodies floating in along with the good news from the blood, right?
No, exactly. And I guess that's the dilemma. If you open up the device, so the kinetics of the insulin the cells are producing that the body needs, and that's fast kinetics, you know, minutes it needs to exert its actions when glucose goes up. If you open it up too much, then the immune system and antibodies can get in and start killing the cells. And that's kind of the dilemma. And if you have too much fibrotic overgrowth, the cells will suffocate. So people are working on trying to get the device vascularized underneath the fibrosis or to prevent fibrosis altogether. Other companies are also working on that. So there are many different approaches, but so far, again, we have not in man. We have seen some promises, but we have not seen a device where you can say now we are ready for large-scale manufacturing, making a device that, for example, can contain insulin-producing beta cells. So, and my guess is it will be a number of years before we are completely ready with that.
And just to jump in from the event organizers' view, I don't think I know any that are engaged in my show, at least. Who are the main device manufacturers and engineers out there at the moment, and what's been asked of them?
Well, I think the reason why we haven't seen any, because there aren't any. I think Bioside is probably the one that is at least furthest along in terms of, there's Beta 2 Air, there's a number of companies, but there's not a good device company like there will be Lonza that can help you to produce the cells. And the reason being is that producing cells from a differentiation protocol is a number of steps, and if you have a tank, you can adapt to that. But making a device, you know, in type 1 diabetes would be one form of the device. Making a device for Parkinson's would be another form of device. Making a device for chronic heart failure would be a catheter that goes through the vein and then into the heart. So each of these requires separate skills and don't have that many commonalities. And I guess that's why that there are not big device companies, at least not yet. And I think they will probably come, but at least not yet. There's not companies that you can go to that can solve all your device problems. I wish there were, but unfortunately it's not the case. So what we have decided at Novo Nordisk is that we, together with our partners who then bring some device expertise, we will try to bring the rest. But it is an area that for some of the indication I have just mentioned, that needs to be built and quite rapidly. Because, for example, if you inject cells into your brain or into your eye, you need to do it relatively precisely. And the better device you have, the more competitive you're also going to be. And most importantly, the more safe is also going to be for the patients.
Mm.
It's complicated, Michael. It's you might be able to contribute something here, Michael. It's like it's like making a nice cup of tea, right? It's like a tea bag. You don't want those bits of tea floating around, right? But you've got to get the flavor out and the hot water in. The old nickname for that Viasite device was the tea bag. And would that it were that simple? Although I'm sure as an avid tea drinker, Michael, you don't think it's necessarily quite so simple, right?
At 8:43 in the evening, I'm drinking something a little bit stronger than tea, I have to admit.
Is that the time? It's always.
No, that's really, really interesting. Well, you know what? You've given me and my team a little project there, and see if when
you're right. You're absolutely right, Michael. It's been underrepresented. There are device engineering teams. They have shown up occasionally in Miami. Ones that, some of them we rate very highly. And there's common themes that, Delfi, it's same as for you. It's all about cell contact, okay? And whether it's the inside of your Prodigy bag set or your Luca pack and the massive tube sets involved there, or whether it's the inside of the tea bag or whatever innovative solution Novo's coming up with. We're not quite sure yet, are we, Jacob? No. Compatibility, just not killing the cells. Again, as Jacob pointed out, whether they're being squirted out of a fine gauge needle into the cerebellum for stroke or Parkinsonism or epilepsy, not killing the precious cells that have been made is just as hard as getting them there in the first place. So I think that is a project, Michael, and it's one that has broad application.
Yeah, and
also, and also, simple issues. I guess that Delfi and GSK also is looking at, how long can the cells actually survive in the container or the device, or just the sheer forces when you are injecting the cells. If you do it too fast, you can actually kill some. Neurons, for example, are really vulnerable to sheer forces. You can kill off the cells quite easily if you inject too rapidly. So it's not just, even if there is an off-the-shelf device, then you need to demonstrate how to handle the cells in that device, or an off-the-shelf bag that can handle blood products, that can maybe also handle your cells. You need to demonstrate that your particular cells then are happy in that container or device. And then most importantly, also once you get to the operating room, if there's operation involved, how long can the cells survive in a steel container, and what if it takes 10 minutes more and the cells goes to room temperature? Will they work as well? It's really, really complex, and a lot of quality assurance along the different steps from when the surgeon have the cells and a device. And then when you get to the pre-filled devices, which probably is going to be the type 1 diabetes device for treating diabetics, as we spoke about, then if you have to do too much in the operating suite, then you need a GMP suite, and then you are limited to very few hospitals, and your distribution chain become really, really complex. And all of these things you need to consider before you basically start your pivotal trials, because they need to be in order. It's very difficult to change later on.
Right. Yeah, there's a lot to think about there. I've got a couple of thoughts in mind of organizations that I I know that are involved in this space, and it's a topic that we rarely talk about on our agendas as well. So maybe there's something there that while we are unable to host events, that we make this we try and find some specialists in this to do some sort of webinar potentially. But thanks for that, Jacob. That's really, really good food for thought. Well, over to Anthony for our last question of the evening, afternoon.
Lunchtime.
Yeah, lunchtime.
Not that I'm hungry or anything. No, I think there's a couple of things we might touch on here. We have to talk about the obvious, which is the pandemic situation. At Dark Horse, we've been monitoring this a lot over the last few months as to how it's impacting our clients, how it's impacting potential future clients. We see this, frankly, amazing tailwind at the moment in the venture capital raises. I think the month of April venture raises in biotech were a full 1/4 of the entire previous year, which is stunning in the face of the pandemic. On the other hand, there's the little issue of a global slowdown, which I think it's going to be almost impossible to escape. Also, we see a tailwind of perhaps, if there's one thing we've learned from this pandemic, perhaps society has learned appreciation of healthcare workers and innovative and focused and fast healthcare development. I think that's a tailwind. And the other headwind is running manufacturing facilities in a distanced manner, even pressure on manufacturing facilities, CMOs and pharmaceutical companies, manufacturing facilities suddenly wanting to pivot to work on COVID-19 related therapeutics and vaccines. I just have to ask both of you, how's the pandemic treating you on the macro level and the micro level in the ATMP world? Delfi, what's it like at GSK at the moment?
Yeah, I mean, in the ATMP world, it's obviously impacted the execution of our clinical trials because everything has slowed down, and it's sort of given us a pause in terms of we have multiple programs, different types of programs, multi-generational programs, as to think if this continues, how we would we wait for newer technologies and not go with the older ones, so it's forcing us to basically take a hard look at our 2 to 3 year strategy. Especially in the area of multi-generational technology development, because we have felt that in the CAR TCR world that that solid tumors essentially present a huge unmet need, and so with that we have thought, well, what are the kind of technologies which will really crack measurable, clinically meaningful efficacy. And we have invested in a lot of next generation technology. So with the pandemic slowing things down, we are thinking, well, does it make sense to do the first gen and second gen and all those things in parallel? Yeah. Or should, series, or should we do it in parallel or just go with the second gen approach? So it's both misses and opportunities. Absolutely.
What's the slowdown like? Are you losing 3 months of trials? Are you losing 6 months? Is it worse than that? Do you see light at the end of the tunnel?
Yeah, I mean, I would say anywhere from like 3 months to 9 months. I've heard numbers across various programs.
Yeah. That's a material delay.
It is. Yep.
Do you think you'll catch up, or do you think that time is lost forever?
I think the feeling is that I mean, we are trying to establish a wide clinical site network to progress our trials. So the feeling is if we don't catch up, we might try to utilize this opportunity to push some next generation approaches which were not clinic ready sooner. So try to make the best of the situation.
Leapfrog. Interesting.
Yep.
Jacob, you're in a more developmental environment than a pre-commercial and commercial environment. How is it at Novo?
Well, I think that I guess the industry we are very similar to what Delfi just spoke about. I think for our stem cell capabilities, we were actually hit initially quite hard because we had a CMO producing cells for our Parkinson trial, and that basically, with day notice, they basically turned the incubators off, and that was it. And now we are back up again. We also have our manufacturing facility in Fremont in San Francisco. That was also basically people were told to go home, and that was also it. They are returning back now. In Denmark as such, we have been very fortunate. I think now we just have 10 people in the entire country on intensive care. We have basically no death anymore. The country is completely opened, and people take social distancing, you know, maybe not the young ones, but most people take it quite serious. So I think as a country, Denmark, Novo is relatively easy hit. We had some, as a company, we were helping the authorities testing for COVID-19 because the testing capacity was not high enough. That impacted us a little bit because we had to put personnel at that. And now we are making antibodies test also to help the authorities, because from a local stand, Novo is quite a big local company. So we are taking that quite serious. And then, we will be facing a few months delay, and we're trying to catch up. So I think, considering the crisis the world has just been through, I think there's really no reason to complain about it. It's a shame for the patients that our products eventually are going to be delayed. But I think looking at what the world just went through, I think we can cope with that.
I think that's a very good way of putting it. Michael, over at Phacilitate, all you do when you're not at conferences, all you do is fly around the world looking at nice hotels to hold conferences in, right? So what on earth? But seriously, how is it at Phacilitate? You're a conference organizer, and there aren't any conferences.
Yeah, I've been trying to reduce my international travel for the last 3.5 years. So, more time at home is definitely one of the positives that have come out of this for me personally. But you're right, though, Anthony. It's, as organizers, the uncertainty of not being able to run face-to-face meetings is really, really tough. Especially when, look, we don't have a direct link to patients that we're accountable for. But our customers do want to get face-to-face, and there's organizations that I know that rely on our events for their business survival, for their opportunities. So
mentioning no names.
Anthony can't even afford a jacket at the moment. Look at it.
Yeah, no, my dress code has fallen off significantly. I think we're all at a point now where nobody is even pretending that we don't miss the face-to-face interactions and the conferences. That the travel's horrible. We all hate the travel, but we love it when we get there. And I've had many conversations. I think I said to you the other day, Michael. We're sort of all confessing that even with the whole being away from home too much thing, just personally as well as professionally, we're really missing it.
Yeah, and the only things I can't replace it like for like. There's a number of organizations that are that have kind of had to do virtual events due to various needs of stakeholders. Fortunately, I'm not in that position yet. But I genuinely don't know what our situation is going to be like come December.
Come December, I'm going to need a hug from Michael at some point. I don't know what we're going to do, Michael.
Yeah.
But hopefully Trump will get it a little bit more under control than he has now.
Oh, yeah.
So we can start meeting again.
Mm.
But I think one thing is the meetings, and there you're right. There the personal interactions are vital, because a lot of the interactions you are having you didn't plan for, and you go hear talks and you listen to people, and I personally really miss that. What I maybe don't miss as much is going a lot of times to San Francisco, to our manufacturing facilities, and I think that there are some changes that are here to stay, that we are going to use video conferencing a lot more than we did in the past. At least I think that's one of the changes that's going to happen at Novo, that we are not simply going to travel as much. I think scientific meetings is an exception, because there the personal interactions and networking is vital. But for business meetings, I think many of them are going to be, and also, you know, we're looking at ways to do process transfers, because we have the process development in Denmark and the manufacturing in the US, to do process transfers, you can say via not meetings like this, but without actually going in person. And there are the HoloLens from Windows and the others that are quite interesting, where you potentially can do stuff like that. And I think that is also going to come much more in the future, and it's just being accelerated right now due to the COVID-19.
You're right, Jacob. We're all learning new skills. We just pushed out a white paper on virtual audits. Yeah. And if you need someone to pop down to your manufacturing facility, I'm just up the road. Okay, so just give me a call.
Yeah, we will.
Good. Any more thoughts on anything else to add around that at all?
I think that's the big issue of the moment, Michael, is we're dealing with this thing, and we're trying to move our products forward. I think Delfi's comments about perhaps cup half full, maybe some products will leapfrog the development cycle, and Jacob, it's great to hear everything that Novo is doing. Novo is a big company, and it's going to be part of the solution, not just in Denmark but also globally down the road.
Yeah, and I really appreciate the fact that we're able to connect on this conversation this evening. And looking at the time, Jacob, I know it's getting late in the evening for you, but I have to say,
my wife's putting the kids to bed and they're still not asleep, so just take 5 more minutes, then it's good.
I don't want you to miss story time. Well,
no.
I hope that's not the 28-year-old that you need to put to bed as well.
No.
Look, this is really, really enjoyable. Thank you so much, guys, for sparing the time to have this conversation. And and thank you to our audience out there listening. I hope you've enjoyed it. We are always eager for your feedback. We're 3 episodes in now, Anthony, and I feel like we're getting a bit of rhythm. But always keen to hear how we can make this show better. So please do let us know what you think. And I always like to end with a little bit of content that we got coming down the Phacilitate pipeline that can keep you engaged. And one thing that, speaking of remote assessments, we've got the European CDMO map is released and has been updated by our editor, Nicola Ambler. So if you are looking for CDMOs on the European landscape, then that is a great bit of content for you to check out and start your assessment. It's got capacities, technologies, and obviously locations and all the latest information about the key players on the continent. So you can find that at the www.facilitate.co.uk/exchange. So without further ado, we're going to wrap up there and let everyone get off. And I really appreciate your involvement. Delfi Krishna, thank you so much for joining us.
Thank you for having me. It was a real pleasure.
Thank you. And Jacob, thank you for joining us again. And I hope the weather gets better for the following 13 days of your holiday.
Yeah, so do I.
Good.
Otherwise, we can do another podcast.
Of course.
Yeah. But thanks for having me. It was really my pleasure.
Thank you. And Anthony, I'll speak to you again soon.
Sounds good. Michael, thanks for hosting.
Take care, everyone.
Bye-bye.
Bye.
Thank you.
Bye
Bye.
bye.